So in this video, we're going to review the lesson that we did last week on meiosis. Okay, and if you remember from that day, we had 5 vocabulary words. 4 of them were new. DNA replication, that was a review word for us. Homologous chromosome, that was a new word for us. Tetrad was new. Crossing over and recombination were new, and disjunction. So these are going to be the 5 words that we use. You don't need to write these down now if you're watching this video for the first time in place of the lesson, but you will eventually. So to get started, I need you to get a couple things out. I want you to get a blank piece of paper with no lines, and I want you to get 2 different color pens. So you need a blank piece of paper and 2 different color pens. I'm going to use blue and red, so if you want to match my colors, you can use blue and red. Okay, so turn your page long ways, not landscape, but portrait, and I want you to draw the circles that we're going to draw meiosis in. If you remember, meiosis has 2 divisions. So we're going to learn those as meiosis 1 and meiosis 2. So I just want you to draw, that's my first division, and then I want you to draw your second division. All right, so we know that meiosis starts with 1 cell and ends with 4 cells, which is something that we're going to write at the end, okay? So the first thing that we know, first vocab word that we're going to go over is one of the new ones. We're going to review that word, homologous chromosome. So over here, I just want to let you know that we're going to have 2 colors, right? You get, as a sexual organism, you get half of your DNA from your mom and half from your dad. So I'm going to draw a blue chromosome, and that's going to be from dad. And then I'm going to use red as my mom color. I'm going to draw another chromosome, and that's going to be from mom. Okay, so we're just going to pretend that mom has blue eyes, so she's going to have a small b, and right there is the gene for small b. And dad has brown eyes, and over here is the gene for big B. Before we review the word, I want you just to write the word homologous. Okay, we know that prefix homo means the same. We went over that the other day in class. And what we're looking at with a homologous chromosome is that they're the same type of chromosome, meaning we have, as a human, we have 46 chromosomes, and we have 23 different types, and they each come in a pair. And scientists give each chromosome a number from 1 to 23. We learned the other day that 23 is our sex chromosome, and that's what determines gender. But 1 through 22 are our autosomal, and they control all of our body traits. And scientists now know where every gene is located on every chromosome. So let's just pretend that this is a certain chromosome pair, and right here on each chromosome is the same type of gene, which would be for eye color. But what the gene codes for could be different. It's still the same gene. It's the gene for eye color. But what the dad is going to give us and what the mom is going to give us are going to be different. So dad is going to give us the gene for brown, whereas mom is giving us the gene for blue eyes, which is recessive, which is the small letter. Big B, small b, something you should remember from middle school. Homo means the same. They're the same type of chromosome, same genes in the same location. Okay, so let's start going over meiosis. The first thing that happens in meiosis, and it's the first thing that happens before mitosis, is it's an interphase, DNA replication. So what I want you to do off to the side over here, so I want you to write DNA replication. Oh, never mind. DNA replication happens in interphase, so let's remind ourselves of that. And it is where these single chromosomes duplicate themselves and become doubled. Okay, that is where they go from single to being doubled, and that is an O. All right, before I go back to the image I'm having you draw on, we need to go over one of the words called crossing over and recombination. You don't need to write this down yet because we're going to write it down in review, but I'm just going to write the word crossing over or recombination. Remember in class we did this with our little construction paper chromosomes. Okay, so let me just show you. In interphase and DNA replication, what we just talked about, we know that these single chromosomes are become doubled, right? So single become doubled. So we have 2 blue, which have the big B's here, and we have 2 red, 2 for mom, right? They are now double chromosomes. They would actually be connected at the centromere. I'm just drawing them spaced out a little bit. Crossing over is when you see a swapping of DNA in the middle of a tetrad. So these 2 pieces of DNA, we're literally going to cut here, and you're going to replace each other and like literally swap DNA. So it's going to look like this. You're going to have your one chromatid that is all father, one that is half the father, and half the mom, so now has the big small b on it. The other one that is half the mom's DNA and half the dad's chromosome. And then you're going to have the one that it's the full mom. Okay? So the tetrad would look like this if it's connected at the centromere. Let me just try to draw this. So we need one more little red piece right here. Okay, we'll just connect them at the centromere. That's what it would look like maybe on another diagram. But I want to make sure that we have ours are kind of spread out so we can draw, oops, right, the chain goes here. Okay, so we have a swapping. And when we look at these 4 chromatids, all 4 are genetically now different. The reason why crossing over can happen in these chromosomes is because, again, they're homologous. So if they get cut and swapped at the same location, you're literally just swapping the same genes. It's not like it's 2 different chromosomes. We're changing the genes when we're working with the gene for eye color. So we are going to assume that this has already happened, and then we can go back to our diagram, and that's what we're going to draw right here. Okay, so let's take your dad color and draw your dad chromosome. And the other one, remember, is half dad, half mom. The other one is half mom, half dad, and the other one is the full mom, which has the small b. Okay? We are going to label this tetrad, and we're going to label this crossing over. Okay, so now it's ready to go through the 2 phases of meiosis. Okay, remember the first division is called meiosis 1. And if you remember from the animation in class that we watched, it has a prophase 1. So I'm just going to put P1 here. It's going to go through metaphase 1. It's going to go through anaphase 1. And it's going to go through telophase 1. I know it's sloppy, but it has all the phases. And it's going to split. Okay? I'm just going to draw a circle here, and I'm going to draw that. Please don't draw this down because I'm going to erase it in a minute. But the tetrad is going to split. Okay? And half of that tetrad is going to go here, and the other half is going to go there. Okay, so let me just get rid of that circle. That splitting of the tetrad is going to be another vocab word we write down later called disjunction. Disjunction is when the tetrad breaks. Oops. So when the tetrad breaks. The tetrad breaks apart. And so this happens in anaphase, just like, oh, sorry about that. It happens in anaphase, just like when the double chromosomes become singled in mitosis. So it's them breaking apart. So now let's draw that. So let's get your dad color out. And over here we have one that was all dad, and then one that was half and half. And then over here we have one that was all mom, and then one that was half and half, but opposite. All right, that is disjunction. We did an online activity, if you remember, when we talked about non-disjunction. So we will talk about non-disjunction, not in this video, but if this goes wrong, if disjunction does not occur, that's how things like Down syndrome and different trisomies can form. So that's something separate that we talked about in class. So now we enter meiosis 2. Again, meiosis 2 has a prophase 2, has a metaphase 2, has an anaphase 2, and a telophase 2. Okay? So that's just going to split these chromosomes into 4 different cells. So let's just draw them. Now get your mom color, finish this chromosome, has half, and then the full mom is over here. So what we do is we've ended with 4 cells. We started with 1, we ended with 4. So now we need to label some facts on these cells, and then we need to go over the vocab. All right, so you have to remember that in meiosis, we're making sex cells, right? That is the function of this process, is to make sex cells. So I'm just going to draw a little flagella on each one of these to remind me these could be 4 sperm cells at the end, okay? A couple words we went over the other day in class were diploid and haploid. Okay, we said diploid is the full set of chromosomes, or we're going to write that on our vocab list, so don't worry. But I want you to label this diploid, okay? And that means it is the full set of chromosomes, so it's the full number of chromosomes. Hold on, diploid, full number. And then we end with 4 haploid cells. So haploid means half. It's going to have 1/2 the number of chromosomes. So if this is a human, this would be 46 chromosomes. We're going to end with 23. Okay. In our little image right here, we started with 2 chromosomes, right? The 2 over here from mom and dad. After DNA replication, they duplicated, obviously, and made more copies. But then once it went through divisions, we started with 2 and we ended with 1. So let's write some facts down off to the side or on the bottom. I'm going to write the facts here that we need to remind ourselves about this process of meiosis. First of all, meiosis makes sex cells. Okay, another word for sex cells are our gametes, which are our sperm and egg. Okay. The second thing we need to remind ourselves is that they have half the chromosome number. Okay, so gametes have 1/2 the chromosome number of that organism. So for example, humans have 46, the gametes are going to have 23 chromosomes in there because it's going to have half the number. Remember, this is involved in sexual reproduction. Okay, well, let's write that. It's used in sexual reproduction. Okay, sperm and egg are going to go through fertilization, which we've already talked about. Now, from the beginning, if you just go up to the top, we started with 1 diploid cell, and we ended with 4 haploid cells. So we need to write that down. 1 diploid cell makes 4 haploid cells, okay, which are our gametes. Now, I want you to look at each of these 4 cells. Look at the 4 gametes at the end, okay? Is there one that is exactly like another, or are they all genetically different? You should say that they're all genetically different. They all have different genes that are in them. So at the end, this is very important, okay? We have 4 genetically different gametes. Remember, that is because of crossing over. But remember, what is the benefit of sex reproduction? Increase genetic diversity. Getting genes from 2 parents alone is going to add to the genetic diversity. But when you add crossing over to it, that means that every sperm cell and every egg cell that males and females make are genetically different from one another. That is why you don't look exactly like your brother and sister, because the sperm and the egg had different genes in them. This just increases genetic diversity, which is that much more of a benefit. Okay, so we're just going to review the words that we will use today. So we're going to start with DNA replication, okay? So just to remind ourselves, now I would actually flip your sheet over to the back if I were you and just make a long list. So DNA replication. We know what happens in interphase. And we know it's when DNA makes a copy of itself. and it's when it goes from a single chromosome to a doubled chromosome. Single to doubled chromosome. So visually, DNA replication is when it goes from a single chromosome to a doubled chromosome. Okay? The next word, and if this goes too fast, please pause to catch up with writing, but the next word we're going to go over is homologous chromosome. Okay? Take your red pen, underline the prefix homo, and remind us that means same. Okay? And it's the same type of chromosome. Okay, remember they come in pairs and they have the same genes on them. So homologous chromosome, same type, same genes. So we have, the one from our dad and we have the one from our mom. They each had the same gene. Okay? These are both the gene for eye color. They each have it. Not every chromosome has that. Only one pair has that. You have a million traits. So you have to have genes on different chromosomes to get all of our traits covered in our genetics. Our next word is going to be tetrad. Okay, tetra means 4, right? So we're going to have 4 chromatids make them up. And this is where the doubled homologous chromosomes bind together. Okay, it's D. And this happens in prophase. It takes a very long time to find each other. It's very different from mitosis. It does not happen in mitosis. Okay, but this allows for crossing over to occur. So crossing over, or sometimes it's called recombination. That is where you have an exchange of DNA between the homologous chromosomes of a tetrad. So you're only going to see this in meiosis 1. You're not going to see this in meiosis 2 because the tetrad is broken. Okay, so let's just draw that real quick. We're going to draw, just if you want to just real quick, 2 blue and 2 red. And this is what they become. Okay, they exchange DNA, which we know increases genetic diversity. I'm just going to circle in yellow where that crossing over actually occurs. All right, and then finally, disjunction. So disjunction is when the tetrad breaks apart. Okay, what this does is it separates the homologous chromosomes. Okay, so I'm just going to have my tetrad here. We just drew that just came from crossing over, and it's going to break apart. Okay, that is disjunction. Very, very important. It is very, very important. Okay. You can put the preview of the word non-disjunction, which is something that will lead to Down syndrome. All right? And that is it. That is the review for meiosis. So you can be watching this video to review for the quiz, or you could be, if you're absent, these were the notes that we took in class.